Well, good evening, ladies and gentlemen. Welcome to the Lakeside Theatre. My name is Professor Graham Underwood. I'm the Executive Dean of the Faculty of Science and Health. I'm hosting the inaugural lecture this evening. So it's a great pleasure to be here and to attend yet another of these inaugural lectures. This is part of a series of special events that the university has been running to celebrate the excellence in research undertaken by our recently appointed professors from world-leading academic departments with international reputations who will talk to you about their research, exchange ideas and hopefully inspire you with what you're going to hear today and make you think about things, taking that away with you. This is the last of the lectures this term, but the series restarts again in the autumn term in October. So research excellence is at the heart of our work here at Essex, and we've established a global reputation as the university over the last 50 years for the high quality of the research work that's been done, and in the least recent research assessment exercise, also the impact of that work outside of the academic community in the world at large. And this year is especially important to us as it is our 50th anniversary year. We're celebrating that over the period of this academic year. And we've heard already this year from professors in the departments of government, sociology, literature, film and theatre studies, history, and also already this year in mathematical sciences. And the themes that we've heard about are in the series of lectures so far have included translating history for television, the role of women in peace and conflict, and in optimization, looking for algorithms from nature. So this evening's lecturer is Professor Ed Codling. He'll give his lecture in a few minutes. Then we'll take some questions from the audience, with Ed and I sitting in our little fireside seats over there. And then we have a drinks reception upstairs, which everyone's very welcome to come and join all of us up there after the lecture and obviously talk to Ed and shake him by the hand and ask him any difficult maths questions you might have accumulating whilst you listen to him. I'm particularly pleased to be here myself to introduce Ed, as I was on the interview panel that appointed him a number of years ago. So, as I say, it's a particularly warming feeling. I feel rather like you're a father or uncle figure, bringing you through. Your father's here, so I don't wish to usurp your post, sir. So let me consider him like a sort of honorary, uncle-y type figure anyway. So it's always really nice when you've appointed someone, you've seen their success curve do really well, and for Ed to get his professorship joining the university 9 years ago is really, really excellent news. So, as I say, Ed joined the university in September 2006 and was promoted to his professorship this time last summer. He'd completed his PhD previously at the University of Leeds in 2003, and then he went off and worked in Ireland at the Irish Marine Institute in Galway, partly linked to the University College Cork. And at that time, he was working in part on models of fisheries management and taking his maths about populations and dynamics into fisheries. And his initial appointment here at Essex was linked at that time to the Department of Biological Sciences as a linked appointment where we have complementary expertise in marine biology. And that's my particular area, so I've sort of followed Ed's career with interest over these last few years. So Ed's main research areas are in the developing fields of what we call mathematical biology and ecology. And that includes two really different but related angles. One is on movement and behavioural ecology. And that's looking at modelling the behaviour, the movement and the dispersal of animals. And that can be from large animals like cows through to microorganisms, and even to single cells in dishes. The underlying principles are sort of common. You can just scale them to different sorts of categories of behaviour and different categories and types of animals. And he's done work on human beings too, and that's received some quite wide press coverage. And then the second angle of Ed's work is around population ecology, mathematical analysis and simulation of what we call in the trade population dynamics, so how populations rise and fall and respond to various pressures. And certainly on the fishing side, that's really trying to manage fisheries in a sustainable way and to work out how much harvesting you can do without destroying your stock. Ed's been really successful in attracting research funding during his time here at the university. He's won grants from the Natural Environment Research Council, from the Biotechnology and Biological Sciences Research Council, from various national government agencies. He's had a link to a large grant from AXA Insurance and also from various charities. So he's successfully funded his research programme over this period of time, and that's contributed to his meteoric rise to professorship. So young. You know. His work's also had quite a high level of impact, and that's really good. His models of fisheries and fish population dynamics has fed through into revisions of the European Union Common Fisheries Policy. That's not the most successful policy of the European Union, but Ed's work certainly tried to make it better. It's usually blocked by the fishing industry, but the scientists do know how to fish sustainably. It's hard to persuade fishermen of that fact. His research on crowd behaviour has resulted in a number of high-profile things. For example, residencies at the Science Museum in South Kensington, a number of weeks spent down there over the last few years, and in a wide range of media coverage, including television. For example, Dara O'Brien's Science Club a year or so ago, and quite recently on South Korean television, which I did just look at the web link, but I didn't follow it through. But we don't know how many hits that's had from the UK, but it just shows the global impact of the sort of work that Ed's been doing. Ed's work does have a wide range of potential applications, but I think he has a rather media-savvy sense of titling things, because he has used titles such as Zombie Lab and Zombie Horde Event, and you imagine that that catches the ear and the eye of the media, and certainly has helped stimulate interest in Ed's work. Ed has also featured in 5 of the Big Bang Science Fairs, which are a national programme of UK young scientists and engineers fairs, including the one we held at Essex in Christmas 2014 when Prince Charles came. And after that, a personal invite for Ed to attend the 4-day Big Bang Fair at the NEC in Birmingham this March. So clearly, as I say, catching the eyes of external people who value and see the interest in his work more broadly. So I'm sure all of this is whetting your appetite to hear from Ed himself. So please, would you welcome Professor Ed Codling to deliver his Professorial Inaugural lecture on the maths of cows and other animals. Thank you.
Thank you, Graham. Okay, so first of all, thanks very much for coming along. I understand it's a Monday evening, so it's not the easiest thing to get to sometimes. So I'm going to start with this picture is quite important. You may recognise the one at the back there, because he's sitting in the middle of the front row as well. So Graham sort of summed up my background. Something you didn't mention, which I think is very relevant, particularly if there's any maths students here. I didn't actually study biology at all formally. I think the earliest I did it was a combined science GCSE, and then I haven't studied it formally since then. Why I'm saying that is because I think something I've been really passionate about, what I'm trying to do, certainly with my undergrads in the maths department, is trying to convince them that maths is hugely applicable across all of science. And I think this is a case in point that I've become now a professor of mathematical biology with the mathematics rather than the biology. So I have obviously learnt some biology along the way, hopefully, but it just shows the importance of mathematics. Professor Daddy. So I want to start off with a couple of stories. I did ask Sam if he could give me permission to do this. So just a different viewpoint. We've had the viewpoint of, obviously, Graham. I'll give you my own viewpoint as I'm going along. But it's sometimes refreshing just to see what, maybe a different viewpoint from the younger generation. So when I was applying, you have to fill in a big form when you're applying for a professorship, and fill in all the details and what you think you've done well and so on, and highlight all these things. So I told Sam that I'd put in the application, and did he think I should get a professorship? And he said, no. I said, oh, why? Do you think I'm good enough? He said, no, I don't want you to be a professor, I want you to be daddy. So I am professor and still daddy, Sam, so you don't need to worry about that. So that was nice. That made me feel good, obviously. And then when I found out I did get the professorship, we told Sam that there's some very exciting news when he came home, and I told him directly. And he was getting very excited about this news that he was going to hear. And then when we told him, oh, yes, I've just been promoted to professor, he's like, oh, I thought you meant there's something really exciting. So that's maybe just one way to sort of avoid getting too caught up in the moment, maybe, when you get promoted. Okay, so I won't go into detail about this. This is a Wordle that was actually generated. This you can go online to do this yourself if you're interested. This came from all the titles and keywords in some of my papers. So these are the sorts of things. The bigger the word, the more important it is, the more times I've used it. So this is a sort of indication of some of the things I've done. What I want to highlight is the fact that it's very interdisciplinary, it's very collaborative. So I've been working with biologists, ecologists, computer scientists, and in fact, there's a selection of them up there. Not everyone I could find photos for. It's supposed to be roughly chronological order, so I'm very pleased to have Nick Hill, who's just sitting down here, who's my PhD supervisor, all the way down to Rick and Hadi, who I've only just started collaborating with more formally recently. And there's lots of guys who I've done a lot of work with, and some guys I've done a little bit of work with. Everyone I'd like to thank, because I wouldn't have got here where I am now if I hadn't had this such good collaboration. So that's what I wanted to get out of the way first. Right, before I start talking about my stuff, I want to do a quiz. So let's see where we go with this. I want you to have a look at the screen here. There's some mathematical patterns. And you'll see that the theme of patterns is going to come up a lot, so we can come back to this later in the talk. Geometric patterns or number patterns, I want you to take a minute just to have a look at those. What's the next one shape or two shapes, or the next one number or two numbers in each of these sequences? So have a look at those, see if you can think. Some of them are easy, some of them are less easy. I'll give you about 30 seconds to have a think. This feels like one of my teaching lectures, actually. I'm getting a bit carried away. Okay, so can anyone shout out what you think the next pattern is there? What's the next shape going to be in this sequence? Anyone? Blue down? Yeah. Next one in this one? 4x4 square, yeah. This one's a bit more difficult. Anyone guess? I can see someone sort of pointing, trying to say, yeah. So number one's a bit easy. This one's a bit easier, so I won't go through that. 5, 6. This one actually is very, very similar to that, so I'll go there. What about this one? 8? 7? 7, 11. Can anyone guess what the next ones are after that? Yeah, so that's, if you're a mathematician, you spot that's the prime numbers. This one, 5, 8. Now, the reason I'm showing you this is because I'm going to argue that actually biology is all about patterns, and maths is all about patterns. And actually, I think they naturally come together very, very, very well. So everyone who's actually got some of those right, you're a mathematician. But actually, if you look in the real world, that pattern, 1, 1, 2, 3, this is known as the Fibonacci sequence. And that's a very, very famous mathematical pattern because it occurs a lot in biology. So you can create a square, for example, of what size 1, size 2, in terms of the lengths, size 3, size 5, and so on. And these match the numbers in the Fibonacci sequence. But if you put them together in this particular tiling arrangement, they fit together quite neatly, but also you can create this sort of spiral if you go around from the corners. And if I show you that picture, this is a shell, obviously, from I don't know which species that is, I should have found that out before. But the key thing here is that the pattern matches very, very closely to what we observe in nature. And actually, the Fibonacci sequence has been used to look at the distribution and the shape and the pattern of the seeds in a sunflower head as well, and lots of other examples as well. So mathematics can certainly explain the patterns we observe in the living world. So there's just two examples. But is this surprising? I would argue probably not. If you think about how mathematics came about, if you go back thousands and thousands of years, people had to come up with a way to count, to measure things. And so they created a system which we now have developed into this formal system of mathematics. But from my point of view, and other mathematicians may disagree, there's an argument that mathematics is completely abstract from the real world. I would argue it's not, that there always has to be some sort of connection to the real world. And then it's not surprising that we then find these sorts of patterns in the living world that we may be interested in. So if you're thinking about the mathematics of life, what I want to think about then is the key processes that living systems all typically are going to sort of exhibit. So the first one is growth. I mean growth in a very general sense. If a population grows, then obviously you've got animals being born, and if the population is growing, there's more births and deaths, for example. But you could also have individual growth. So this is sort of development. An individual grows bigger, so you've got a small lion cub will go into a big lion cub, and so on. So these things are patterns over time. So the population as a whole is changing over time, so going up and down, or an individual animal might be growing and getting bigger, so it's going forward in time. The other key thing that I'm generally interested in terms of modelling living systems is movement. So Graham mentioned that I did a lot of work on modelling movement. So this is examples like dispersal, migration of birds, foraging, and also social movement. So this is where some of the crowd behaviour things we've been looking at. And again, these things change over time, but particularly now we're also looking at space. So if we just have a population of animals, we don't necessarily know or care where they are in space. But if we're looking at movement, we do want to know that. And these two key processes are what I'm going to talk about a lot later in the talk. So it's all about this sort of interaction, population interaction, growth, reproduction, so the changes, dynamic changes in time, and the movement, the changes in space. Now, I know there's plenty of biologists in the audience, so if I try and argue that these are the only two key processes for living systems, I'm sure I'm going to get a lot of criticism. Feel free to criticize me later. I'm aware there's lots of other things, but these are the two I want to concentrate on for reasons hopefully will become clear later. So the first one we'll look at is this sort of temporal pattern, so patterns in time. And the classic example is the Lotka-Volterra model. So this is a model that came about to model predator-prey cycles. So in other words, interactions between a population of prey and a population of predators. So a famous example is the Canadian lynx here and the Arctic hare. And why they use that is actually they've got a very long time series of data from when they've actually been tracking and looking at the animal pelts that were collected in Canada. So this is the first bit of maths we'll show. If you're not a mathematician, I don't want you to get confused by this. All it's saying is that this is a differential equation. It just says the change in our prey population. N is our prey population. This is just how much it's changing. On the right-hand side, that change is equal to this term here, and that's just the growth. So A is a positive number. And our population is just growing according to the amount there is. So if you've got a big population, it will grow a lot because they all presumably reproduce. If the population is very small, there won't be a lot of reproduction. But there's this term which is taken away. This is a predation term because it depends on the amount of predators, P, and the amount of prey you've got. So if there's lots of prey, lots of predators, you'll lose a lot of the prey. On the other hand, this is the change in the on the predator population. They grow by a certain amount, which is related. Notice this term is very similar to this one, but it's positive. They grow by the amount which is related to the amount of prey that they're eating themselves, which is what you might expect. And then there's a decrease in the predator population, which is on the assumption that obviously if they don't find any food, there's a continual decrease because they're going to die of starvation and so on. So these two equations are known as a coupled system because they're linked together. As we change N, the prey population, automatically the predator population will change. So then what we get, if we imagine running that forward in time, the prey population starts to grow, and then what happens, because there's lots of prey, the predators go crazy. They've got loads of food to eat. They grow as well. But you get to a point where the predator population somewhere here has got so big that the prey population suddenly crashes because there's so many predators eating them, they drop down very quickly. But notice there's a delay because typically these lynxes, they live for quite a long time if they have food available. So they can probably live for a few months when there's no prey and living on their fat reserves and so on. So there's a delay before they crash as well. And then because they've crashed, the prey go crazy and they grow. And it cycles around like that continually over time. So very, very similar systems apply in, as Graham mentioned, fisheries. I did have about 7 slides all on an example of fisheries, and I realized I was going to run an hour over time, so I cut them out. So that's the example, a very simple example of predator-prey. So we mentioned movement as the other key thing that one of the things I'm interested in. So this is a very abstract sort of mathematical description of movement, which is known as a random walk. So the example that is easy to sort of visualize is maybe, I'll act it out. Imagine I tossed a coin. If I got a head, I'm going to move to the left. If I get a tail, I'm going to move to the right. And it's equally weighted coin, so assume it's 50-50 chance. So this probability of moving left is the same as probability of moving right as a half. So I might want to think about, if I stood here and did this 20 times, what would happen to me? Would I sort of go backwards and forwards and end up pretty much where I started from? Or would I sort of just go all the way off the edge of the screen? The cameraman's going to hate it. So you might ask that question. We can look at that mathematically. And this is what we've done here. So this is just sort of crunching through the numbers and working it out. So what we're saying is after one step, or in fact after 0 steps, I'm where I started in the middle here. After one step, I then toss a coin. I'm either going to move left or I'm going to move right. I can't stay where I am because I always just move. We can carry on that through. So then after so many steps, I can work out the probability, for example, that I'm 5 steps to the left or 5 steps to the right, or whether I've come back to the middle. And that's known as a random walk. And so I did a whole PhD with Nick on slightly more complicated random walk models, because obviously that's not particularly realistic if you're thinking of animals moving around and so on. But you can then plot this out. This is sort of a path if we were moving backwards and forwards. So imagine me moving backwards and forwards, tossing my coin. This is what you might look like. These are just 5 realizations of a random walk. What I want you to notice and take away is that we start off at the origin here, but very quickly we start to spread out in space. So that random walk, you can actually model that mathematically. I won't go into all the details, but if you've done my mathematical biology course, you'll have seen the details. In fact, my students have an exam tomorrow, so hopefully this is good revision for them. So this is basically the equations that describe what's going on. And this is a visualization of this solution as it's looping around. We start off with a quite high peak around the origin. That's where all our individuals are starting. But then they spread out in space. So they're spreading out. So imagine if not just me, but lots and lots of people with me, we all started doing that coin-tossing experiment. We'd spread out across the stage eventually. We wouldn't all stay. Most of us would remain around the middle, but we would keep spreading out over time. We can do this in 2D as well. So we can imagine a random walk now in 2 dimensions. So now I toss a coin, I toss 2 coins, one to go left or right, one to go backwards or forwards, and I'd move around the stage here maybe. Again, we have a diffusion equation which describes this movement, and this is the solution. This is actually not quite the solution of this because I've got an extra term which is moving me here. But the key point to take away is that I start off with a big peak and start to spread out. So this random movement, this diffusion, when it's only there on its own, it spreads this population, all my individuals I have, out in space. So diffusion acts to get rid of patterns. If I have a big spike at the start here, where everyone's stuck at the starting point, diffusion will act to sort of smear it all out over space. So it gets rid of patterns, diffusion. So that's the key thing to take away from this. Diffusion on its own, because I'm going to come back to that in a moment. So we've talked about predator-prey dynamics, for example, the lynxes and the rabbits, or lots of other examples you can probably think of. We've talked about movement. But we might also be interested in how those two things link together. And that's known as a reaction diffusion system. So reaction, because there's some sort of interaction between maybe 2 species, and diffusion, because we have movement in there as well. And this is, reaction diffusion models are used a lot to model biological systems, spatial biological systems, because obviously it's, as I said at the start, two key processes for these living systems. So this is, I'll go into this in a bit more detail in a moment, but this is basically a map. Can anyone recognize where that is? Anyone spot? If you look at... Yeah, so that's Florida. It's the Gulf of Mexico. This is basically tracking a plankton bloom that was around the time of the oil spill, I think a couple of years ago. And basically what's showing is the red is high densities, high concentrations of phytoplankton, which is the key species if you're modeling or looking at marine systems. And blue obviously low, but notice that there's a lot of sort of clumps and patterns of sort of medium density of phytoplankton and a few very high dense patterns sort of coming down here. Obviously, there's lots of stuff going on here. There's lots of circulation. There's lots of things going on in the ocean and interactions in these plankton species. But what we can do is simplify it down a lot. And let's suppose we're just interested in phytoplankton, which we'll call U, and they're the prey. And we've got microzooplankton, which is a predator, and we'll call them V. So we've got some equations which describe what's going on here. And again, don't get confused by this maths. All this is saying is the change in our prey population has got some diffusion in X and Y, so it's 2-dimensional. And then this term basically says we've got some growth and we've got some predation. That's all it is. So it's just balancing what's going on. Our predators, we've got some change in the predators, which is due to diffusion. And then we've got some growth, which is related to how much they eat the prey. And we've got some decrease. Very, very simple system. As I say, the real world has got lots of circulation going on, ocean currents, which we're not modeling. And in the real world, we obviously have higher species. We've got fish and things. And we're not including them in this model. But even a very simple model like this can produce some very interesting patterns. So this is a video produced by Tahani, who's my PhD student. I can't see anyone in the audience, so hopefully she's there somewhere. And this is just evolving this system over time. So we're just running it for the time units here, I think, correspond to hours. So it's a few hundred hours. Notice what starts to happen. We started with a very uniform pattern. But very quickly, these interactions have basically led to what we call spatio-temporal chaos, in that you can't predict the patterns that are going to appear. And if we let it run for a bit longer, you notice the patterns start to look very, very similar, at least qualitatively, to these patterns that we get here. And as I said, this is a very, very simplified version of this type of system. There's a lot going on here, which we haven't captured in here. But even so, this still gives quite interesting qualitative patterns. If I leave this playing, everyone's going to be hypnotized, so I better go off that slide. So what we've said so far is diffusion and reaction diffusion either destroys patterns or it creates these chaotic patterns. So some of you may recognize Alan Turing. He's the father of modern computing, people say, and he certainly made a huge impact. What people don't know so much is he also pretty much made, he didn't quite invent mathematical biology as a subject, but he certainly made a massive impact on it in his spare time, effectively. So this is, as a mathematical biologist, it's always quite disconcerting when you realize you spent hours and years of your work trying to produce all these good papers and so on, and he did this as a hobby in his spare time, and he actually completely advanced the field. Anyway, what he did was a paper called The Chemical Basis of Morphogenesis, and what he was interested in was how animals or other interactions, when morphogenesis is when a biological system grows and takes shape. So if you think of a ball of cells as an embryo might be developing, how does that embryo form a shape, essentially? Now, he looked at it from a chemical point of view. So he was interested in how maybe in the womb, maybe there's some chemicals interacting. At what point do those chemical interactions mean that the sort of developing embryo starts to develop a head or arms or so on? Or we'll look at in a moment, in a few slides' time, how animal coat patterns, for example, might form. So these equations, although they describe a chemical system, they're still very much the application to a biological context. And what he started with, again, is a reaction diffusion system. So we've got some reaction terms, so some interactions between these two chemicals, C1 and C2, and we've got some diffusion. So this is only in one spatial dimension here, so we're just spreading out along the line. And that's it. It's a very simple system, so we could look at a very similar example to what I had with the plankton. Well, this is actually in a very generalized sense. So this actually, you could convert any sort of reaction diffusion system into this, what they call a linearized type. But what he showed, and this is where he got this from, I don't know, because if you think about diffusion, everyone expects diffusion to, as I say, destroy patterns or create chaotic patterns. What he showed was that under certain conditions for our parameters in our system, so don't worry too much about these details, but the key thing is that you can look at how your chemicals are interacting, how they're moving and spreading in space, and look at how all those things are related. And there are certain conditions, not necessarily easy to get, but there are possibilities where you can get essentially diffusion forming patterns. So think of that's quite counterintuitive. Diffusion, we've looked before, either destroys the pattern or creates this very noisy, chaotic pattern. Turing showed that actually it can create regular, stable patterns. So what he showed was that if you didn't have any diffusion in this system, so if you just got rid of these diffusion terms, there's no pattern. So basically, there's no pattern at all if we don't have diffusion. When you put diffusion in and you have these conditions being satisfied, you can get patterns forming. So what's going on? Well, basically, when you go back to the system we had here, if you have these conditions, what you're sort of forced into, your solution, when you solve that mathematically, or at least one of the solutions, you're forced into a system that ends up with having, so the red here is chemical 1, the green here is chemical 2, and what I'm plotting is the concentration or the amount as we move through space. So what starts to happen here, at the start, so this is the end of, imagine a test tube going, in fact this is an infinitely long test tube, but we start at one point here at the origin, and we've got a lot of chemical 1 and not much chemical 2. And then chemical 1 goes down, and where I start to go down here, I get chemical 2 increasing. So it's a bit like the predator-prey thing, except these are almost exactly coupled to be opposite. So where I've got high green, I've got low red. High red, low green. Now you can imagine if these chemicals actually had a color, then if we started looking at this test tube, when you see these, so this would be what would happen after you'd let the chemicals mix for a while. And obviously what you might expect to see is where there's high red, it appears red. Where there's high green, it appears green. And you get these bands or sort of stripes of patterns forming. This can be shown experimentally. What was really quite fascinating that Turing showed was that he came up with this mathematically before people had actually realized that these types of chemicals might be real. And he actually made a prediction about these chemicals. I think it was one of the first examples where the mathematical theory predicted something they hadn't found yet in biology. So that was quite exciting, quite interesting. So this is, imagine a 1D pattern. So this is just looking along a test tube and you see these bands of color. So diffusion, in this case, caused this regular, stable spatial patterns to be formed. So it's slightly very counterintuitive result at the time, but it sort of makes sense if you go through all the mathematical detail. This is something we'll come back to in a moment. The complexity of the pattern depends on the space available. So if we go back, if my test tube was only this long, and I cut it off here, for example, then I wouldn't have a very interesting pattern. I'd just have one stripe of red, one stripe of green. In fact, if I cut it off here, I just have red. So the more complicated pattern, in this case, it's just bands of colour, but it looks more complicated the bigger the space I have. That will become a bit more clear if I now extend the idea to 2 dimensions. So imagine now we've got exactly the same sort of system, but now we have a rectangular domain. Rather than just this line, which is my test tube, I've now got maybe like a square Petri dish or something, and I'm mixing these chemicals up. What Turing showed was that if you have a very small area, then you can't get patterns forming. Essentially, if I go back, what you need to do is have your boundaries fitting in a certain number of complete wavelengths or half wavelengths. So you need to fit in these complete sort of up and down cycles, otherwise the pattern won't form. So obviously the point is, the bigger your space, the more complete waves you can fit in. And this is what's happening here. We've got this small rectangular shape, and we can't fit any waves of our solution in, so we can't get any patterns forming. Then we might make it a bit bigger, and we've got now basically the wave going, creating a vertical pattern. So we've got stripes. Well, the stripes are horizontal, but the pattern, if we go up vertically. Or we might have it a little bit bigger, the pattern flips around, so now it's going, so we've got, the pattern moves along horizontally, so we've got vertical stripes. Or we might get another band coming in now. So this is looking very similar to the example we had before. But this is 2 dimensions. So as is shown here, we can get patterns going this way and patterns going this way. So the simple patterns, early on, we've got not much space. They're all going to be striped patterns. If you think about it, they are the simplest patterns. If I just split this area into 2, that's the simplest pattern. If I split it into 3, again, I'm always going to get stripes. But as we go a bit bigger with our sizes, we can get these sort of checkerboard patterns, because I'm now be able to fit these complete waves in going vertically and horizontally. So you imagine like a chessboard pattern coming out. So the bigger the size, the more complicated my pattern can be. And of course, this depends on what chemicals you've got, what particular context you're thinking of, but this is the idea behind how these patterns might form. So we looked at rectangular domains there. You can, of course, have circular domains, and you can get these interesting sort of radial patterns where you sort of get these concentric rings of patterns, or you get these little dots coming in. So this is, on the right-hand side, is a mathematical model, a mathematical simulation, and these are real fish patterns. So the point is that the mathematics can capture and possibly explain what's going on in terms of how these fish, the patterns form on the fish coat or skin. So if anyone read the postcard that came out from the sort of blurb for my talk, I would explain why leopards don't have spotted tails. That's a leopard. I sort of take it back a bit because they do have a bit of spots on the top of their tail. I'll rephrase the question. So it's, why do leopards and other cats have stripes at the end of their tails? So I'll sort of rephrase it slightly. I realised I perhaps over-egged it a bit. So this is, at the top here is a cheetah, leopard, and this is a genet. They all have stripes at the end of their tails. So these are pictures. They have stripes all the way around for the genet. This is a jaguar as well. I haven't got a picture of a jaguar. Notice on their main part of their coat, it's quite a complicated pattern, the spots. Again here. On the tail, it's stripes. So there's a guy called James Mory, again, who's another well-known person in mathematical biology. He went away and actually did some computer simulations. Rather than having a sort of boring circular domain or a rectangular domain to run his sort of mathematical chemicals and see how they interacted, he did a computer simulation where he had this basically a tail shape. And then he ran these simulations and just to see with different chemical interactions what patterns might form. And as you go from A to B to C here, essentially it looks like they're the same size, but actually this is scaled up by a factor of 10, I think, on each case. So this tail is actually much bigger than this one. And this tail is slightly bigger than this one, which itself is bigger than that one. But as the tail gets bigger, the complexity of the pattern that can form is obviously more complicated here. But even in this more complicated case, as you go down towards the end, you get the stripes occurring as well. So as you move down to the end of the tail, because the spatial domain gets smaller, the pattern gets simpler. So that's basically the explanation why if you've got a cat at home, or you've got any other animal, think of a zebra, for example, it's got stripes. If you look at a zebra's tail, it will be plain. So an animal will always have a simpler tail in terms of the pattern at the end than it will have on its body. That's just a fact. So I promised Sam that I'd have a picture of a Star Wars person in here, so I dig this one out. It does fit, hopefully. This is Ahsoka. She's one of the characters from Star Wars Clone Wars. She's got this thing called a head tail because she's an alien, so she just does. But she's got these striped patterns. Now, if you look at her skin color, these are tattoos, so her skin is actually completely plain. So I would argue that's perhaps either we know when Turing and other people, Jim Mory, who carried on his work, there's something going on maybe on these alien worlds, or maybe there's some slight scientific inaccuracy in Star Wars. I'll leave you to make that decision. Okay, but Jim Mory as well, he also, I don't know where he got this idea to do this, but I would love to have come up with it myself. Not just simulating tail-shaped domains, he actually created a computer program that would create this sort of squashed animal, which looks like an animal carcass, but animal skin, and then he ran some simulations of how these mathematical patterns would form on these types of animal coats. So again, what's going on here, I don't know if you can see the numbers here, but this is essentially a scaling. This is 0.1 down here, 0.5. And it shows how the size increases. So this is very small. This is getting a bit bigger, and the size increases as we go through here. In fact, this one is 5,000 times bigger than, in fact, no, it's 50,000 times bigger than this one. So what notice happens, you start off with a very plain pattern, which again corresponds to this very simple, we can't get these complete waves in the domain shape, it's going to not form any patterns. And then we get it split into two. So it's just like a very, very simple black-white. Again, we had some examples of that earlier. Then as the size gets a bit bigger, you get these slightly more complicated sort of twisted striped type patterns, or these sort of splotches. And then what seems to happen as well is you go even bigger, the pattern almost gets so complicated that certainly if you're looking at it from a distance, it almost looks plain again. Now again, this depends on exactly how your chemicals are interacting and what the sort of assumptions are in the model. But from this, we might predict that, for example, small animals are more likely to have plain coats. Animals which are sort of somewhere in the middle might have these more interesting markings. You think of sort of animals the size of leopards or cheetahs or so on. And if you get a very, very big animal, you might expect, again, to what looks like a very plain pattern. So you think of elephants and whales and so on. That's not necessarily explaining what's really going on. It's just a possible prediction if this model is accurate. So this was Murray's mathematical simulations. So you might think, well, hold on, you don't really get animals that look like that, do you? In fact, you do. So that's a Swiss mountain goat. They literally are black and white and two-tone goats. And remember I showed you the slide where you could go horizontal or vertical stripes? This is a honey badger, again, where he's got his stripes now going vertical. Literally, if he puts his tail out straight, it's literally half and half. Or we've got these sort of bands. So you've got white, black, white. And this is a tamandua, a particular species of anteater. And of course, if you can still see in the background here, these types of patterns here, where there's somewhere in the middle, are very reminiscent of the classic Frisian cows. So this is the hint for those of you who are supposed to be having a hint. The rest of you will see what that means in a moment. So this brings me on to the maths of cows. So I've talked about patterns in terms of processes, these animals interacting, predator-prey, over time, and also how they might spread out in space, and also how those two things then interact to create these interesting spatial patterns. What I'm going to talk about with the cows in a moment is patterns of, again, time and space, but also patterns of behavior. So before I go into that, I want to talk a bit about the sort of context, why we're doing this. So this is a project with the guys who are on the middle row there. If you want to wave. So Riddle College and a few other guys who are helping us. We're interested in trying to come up with methods to try and automatically detect illness in cows. Now, why is that important? Well, these are the sort of gruesome pictures that John, I think, has provided me, a colleague from Riddle. So this is where a cow has got either an infection or probably some cut or damage on the hoof, and that causes problems. So cows that have problems with their feet, they don't, well, obviously they feel poorly, they can't walk very easily, so this cow is basically not putting weight on the back leg here, because this red line, the cow natural shape should sort of come up and then drop down and sort of come up at the shoulder again. So this cow very clearly is not walking properly. And it costs the farmer money if they're not producing enough milk, if they need treatment. The other main disease they have, obviously, when they're producing milk is if they get an infection of the udder, get mastitis, that costs lots of money for the farmer in terms of lost milk production and getting the treatment. So there's a need for early detection of disease on these farms. So you might ask, well, why not just monitor the cows by having someone follow them around all the time? Well, obviously, that's very cost inefficient. So we want some sort of automated system where we could maybe automatically track them and automatically process the data that's coming out and then try and make predictions about their health. So that's exactly what we're doing, funnily enough. So this is a picture of a barn. In a moment, I'll show you some pictures of what these things look like. But basically, you've got cubicles here. These are the pink things. This is where the cows lie down. In the middle here is the feeding passage where they get food. And that's pretty much their environment. There's other things like a scratching post and a water trough. And that's pretty much what they do on a day-to-day basis. So this is the feeding passage, cubicles where they lie down, water trough, because they produce so much milk, drink huge amounts of water every day. And their most probably exciting entertainment is going and using the scratching post. If you ever go to a farm, this scratching post looks like it would be quite fun to run your hand over. It's actually really, really, really sharp and sort of scratches your hand to pieces. So don't do that. But the cows love it. So what we want to look at is patterns of behavior, as I said, in space, both space and time. So not just looking at the patterns in space, essentially pretty pictures. We want to try and interpret that and see what it means for the cow's behavior. The point being is that we think, or we expect, that ill cows are going to behave differently. So without sort of overgeneralizing, if we're feeling ill, we're not as active, we don't maybe socialize as much as we would do normally, and that's the basic idea that we're trying to explore. So we're using a system that basically creates a wireless network. So we put these sensors on the cows, and they have a collar, so the sensor's on the back of the neck. And that sensor, it can detect movement. It can also detect basically the spatial location. So if you know a GPS system where you have a sensor that talks to a satellite and you triangulate by getting signals from several satellites and work out where you are, our system works by essentially all the sensors talk to each other. They work out how far away they are from each other. And you can basically do a big triangulation problem and work out where every sensor is in relation to each other. The good thing about these is they work indoors. GPS wouldn't work in the barn that we're using because it's got a big metal roof. You wouldn't get the signal out. So the big data set that we haven't really done much exploration of yet, but we've got 150 cows which we tracked for 6 months at 1/8 hertz. That means we're taking one data point every 8 seconds. That data point has their location, how much movement they've been doing, and other things about the direction where they're facing and so on. So we've got huge amounts of data that we've been looking at. So I'll talk a little bit about some of it in a moment. What I want to do is an interactive demonstration, though. This is potentially going to go horribly wrong, but we'll see what happens. So this should come on now. Oh, yeah. Right. So I've had some plants in the audience who have been given some different instructions to do. So one person was told to keep the sensor completely still, even though they're holding it. I want the audience to try and guess which sensor it is. Can anyone spot? There's one sensor, or there's 3 sensors here, which are just in the box. They're not being used, so I can tell you that. So any offers for the person who was told not to move it around very much? Top right? Yeah. So the person who was told not to move it around very much, can you check your code and see what the code says?
Sorry, I was asleep.
All right, that's exactly right. Yes, there we go. So we've predicted, or we'd be able to spot from the pattern that emerges what that behavior was. So someone was told to shake it up really, really, really active and make lots and lots and lots of movement. Can anyone spot who that one was? In fact, there's 2. I'd probably say that one or that one. In fact, I know it's that one. Do you want to stand up and wave your sensor? Yeah. So there we go. Who was 2BE? Yeah. So you were told to do it 5 seconds on, 5 seconds off. So you can sort of see that it's up and down, whereas Sam, when he was shaking it up, lots. Graham was told to do it 10 seconds on, 10 seconds off. So you notice there's up and down movements here. So the point is that there's patterns that we're spotting here, and we can detect that with our sensors automatically detecting. This is just the amount of activity, the amount of movement that our sensors are detecting. Of course, we've got more information than that. We've also got spatial location, and we can do a lot more with it, which I will explain in a moment. Okay, so that's the basic idea. That was like 2 minutes worth of, we took 3 hours to set all this up this morning, so to get this right. Can we go back to the normal? Yeah. Okay. So when we do it with our cows, exactly the same sort of signal for the accelerometer, that just measures the amount of movement. This is a cow that's lying down. So again, notice there's not a huge amount of movement. And the cow that's feeding, and think about where the position of the sensor was on the neck. So when it's feeding, it's doing a lot of up and down movement. So of course you'd expect this regular, and you can just about spot it, the up, down, up, down, up, down. Oh, that is quite noisy. And then it was lying here. Notice that this isn't quite as a simple and clear flat signal as the other period of lying. And because the sensor is on its neck, often if a cow is lying down, it might twitch its neck. And of course, you get a big spike of movement. So a lot of what we have to do in terms of the mathematics is take these signals and then try and interpret them and trying to avoid these sort of errors or false signals that we don't want to try and place too much emphasis on. If this was just a cow twitching, we don't want to suddenly say it got up for 2 seconds to feed and then got down again. We want to try and interpret that a bit more correctly. And then this was the cow was feeding again. So in the middle here, this was actually the observation that I think it was probably Zoe, who's somewhere in the middle row, I think there as well. She saw that between these time points, the cow was lying down, which corresponded to this. Then it was feeding, then it was lying down. Now, clearly, obviously, with an observer watching the cow, we know that the cow was really lying down here, even though that signal looks a bit, like we're not quite sure whether it's really lying down or maybe it's moving up, moving a little bit. What we can do, though, is, I won't go into all the details of the mathematics behind it, but there's various ways that we can take this raw data, this signal, and basically crunch the numbers that come out to try and make some sort of prediction about what the behavior is. So this output on the bottom line here is what's come out when we've plugged this data into our mathematical model, our algorithm, to try and predict what's going on. And so between these time points, our model is predicting that it's lying down, so it's got it pretty much perfect. And then this period of feeding, it's got a little bit wrong here where it said, just for a couple of seconds, it says it stopped to lie down, which is probably maybe these low points here. Of course, what we can do is filter this information. We know a cow is not going to lie down for 2 seconds in the middle of feeding, so we can probably filter that out. This bit here is a bit more problematic because these are big spikes of movement, and again, they're sort of shown here. We'd have to think very carefully about whether that really does mean something or whether it is just noise. So, for example, a cow that's actually ill might be getting up and down, so standing up and lying down, a lot more than we might otherwise expect. That might be one symptom that it's demonstrating. So we don't want to necessarily filter everything out because it might be important signals. And that's where a lot of the work we've been trying to do is sort of adapting and improving the algorithm so that it fit with actually the biological reality. Because remember, what we're trying to do is an automated system that detects when these cows are ill, but without, obviously, we've got to balance it between getting it too sensitive so that every single thing here is flagged as something that's maybe different and we have to go and check the cow, or smoothing all of it out and not really picking up the key behaviors that we might be interested in. So the other thing that can help us distinguish between those types of behaviors is the spatial position. So that was just the accelerometer, the amount of movement we had. But this is, I'll show you in a moment where this is in the barn. Why I'm showing you without it is because if we just have this trajectory, a lot of my colleagues tag wild animals and just get trajectory data, so in other words, the movement path. And then they try and interpret what the animals are doing. Of course, we've got a lot more than this trajectory. We've actually got a lot of details about the environment the cow is actually in. So if I just stop it here, this was the trajectory. We saw how it sort of generated. They came over here, stayed there a bit, and then went over here. Think to yourself, what might that cow actually be doing? I showed a picture of the barn earlier. I will show you in a minute, but just try and think what you imagine that might represent, that movement path. So in the middle here, we've got that feeding area. Here is where all the cubicles are. There's some passageways which I haven't sort of marked in, but this is the area where they might rest. And this at the bottom here is the passageway where they come into the barn from milking. So again, hopefully now you can see when we link the trajectory or the movement path with the environment, we can work out probably what the cow's been doing. So it looks like it returned from the milking parlour here, went over to the feeding area, so it went all the way over here, spent a fair bit of time moving around in the feeding area, probably finding the best bits of food and so on. Then it went and stayed here. It's probably lied down in a cubicle. It's probably not moving around all this time. There's a little bit of noise in our system because there's lots of metal in the barn. The signals from the sensors get bounced around everywhere. But this is probably a cow lying down. Then it moved, wandered off over here. This is probably the edge of the cubicles, and wandered down. Notice what it did here. It came down to the feeding area, decided it didn't like this area where it found itself, and moved back to where it was feeding before. This is what we call the sort of site fidelity, the region of the feeding area it goes back to. It was consistent there. And that's something actually we didn't think previously would be that important, but with a bit of exploration that we were looking at recently, we think that actually might help us distinguish between ill and healthy cows. So this slide here hopefully illustrates it a bit better. So this now is essentially a heat map, a plot of where the cow has been over a full day. This is basically just adding up all the periods. So if you imagine here you'd have a big high spot because it's spending a lot of time there. Here you'd have a big high spot. What we've essentially done is done the same thing here, but just sort of coloured it in. So this is an area where the cow was a lot, and so on. So this cow, over 2 days, was fairly consistent in its location. So it's got what we call high fidelity. There's low variability in its space use. It's sort of using the same area as the barn all the time. This cow was sort of just all over the place. There's almost no consistency between the 2 days. This is just 2 days, 2 cows, but this cow was healthy. It had a lot of variability, not very much fidelity. This cow seems to be not doing as much. And we think this might be there's a few issues with the data that we're running into at the moment. We think this might be a real effect. And whether it's effect that because the cow just feels a bit, bored and because it's ill, it just wants to do the same thing. So it's actually a behavioural thing, or whether it's more indirect in that it might be, for example, that the cow here has got a cubicle that none of the other cows want, and where it's going to feed is where it's the best food might not be, in which case it's sort of the other cows might be forcing it out. It doesn't want to fight for the best locations because it's ill, so it might be an indirect effect. But it does seem to be a pattern that we weren't expecting to see that might help us identify either, in this case, lame cows or healthy cows. We can put that information together and look at space use over time. So this is linking the space and time again together. So just very, very quickly, all I want you to sort of spot here, the red cows are the healthy cows, and the black cows are the ill cows. I should have done it the other way around, of course. But if you look over time, what seems to be very, very consistent is that the healthy cows spend less time lying. Notice the red line here is almost universally below the black line. But they spend more time feeding. The red line is almost, apart from here, universally above the black line. So again, that's something we think is going to help us distinguish between these healthy and ill cows. Just to finish off, I want to talk also about social networks. So this is actually quite similar to the social networks that you guys probably indulge in, Facebook and so on. These cows hang around with each other. They have friendship networks, believe it or not. And in fact, they can get quite stressed when they're not with the cows that they like hanging around with. In social networks, when people look at animal groups, typically what they do is just look at what they call proximity or association analysis. So this is basically, are they sitting next to each other? Well, not cows. Are they near each other at a particular time of the day? So if you guys, for example, if you turn to the person sitting next to you, is that your best friend? Probably not. But this is what a lot of people do when they do these analyses. When they look at wild animals, they'll sort of take a picture or they'll track them like we use in the census we've got. And if the animals are next to each other, they'll say, right, that counts as a sort of friendship interaction. But of course, as we can see here, just because you're sitting next to someone doesn't actually mean that you're remotely friendly with them. It might just completely random interaction. So we did a small trial with 7 cows in a small contained barn just to see whether we could get our system working with them and see whether we get any interesting results. And basically, this is just how many encounters they had with each other. So obviously, it's symmetric, because when cow 1 meets cow 2, obviously cow 2 meets cow 1, by definition. And we can construct this network. Basically, this just shows how many interactions there are between these cows. So where it's a thicker line, they had more interactions. So from this, you might ask the question, which cow is the most or least social? So it looks like cow 5 has a lot of interactions. Maybe cow 2, perhaps, or cow 1 is not quite so social. But we were quite, we're skeptical about this type of analysis, because just because cows are near each other, or any other animals near each other, doesn't necessarily mean it's a positive interaction. And in fact, cows, they may actually have very, very negative interactions. They can block each other from the favourite cubicle or from the feeding area. And they may be next to each other, but it might be very, very sort of passive-aggressive type of action. So it might be quite a negative thing that we just don't know if we just use proximity. So what we tried to do is look at what's known as approach and leave vector. So we looked at now these associations where these cows were seen together. But what we did is went a few time steps back, because we've got that data, and we saw which cow actually approached the other one, or did they just sort of randomly come together? And then similarly, we can move then forward in time and see which cow left the other one. So what I want you to have a look at in this picture, it's very difficult to interpret all this, but I'll just very quickly go through. This is how many times cow 1 had an interaction with another cow. And of all those interactions, this is how many times we could identify that it clearly approached another cow. So out of 1,000, or 1,100, it clearly approached another cow 250 times. This is how many times another cow approached it. So look at the numbers there. This is how many times this cow was seen to leave another cow, but without just classifying who left who. When we look carefully about who left who, this cow never left any other cow, ever. But 517 times, so half the time, another cow left it. And look, in contrast, so these are the ones I want to highlight, this cow left other cows 227 times. No other cows ever left it. So I showed on the previous slide, we thought maybe cow 2 was the least social. Actually, cow 1 seems to be the one that's very odd, because it seems to be going after the other cows a lot, and the other cows just desperately trying to leave it. So is that dominance, sort of hierarchy effect, or is it just these sort of needy cows? I think in reality, actually, what happened, this cow is a sort of a runt cow. It's half the size of the other ones, and it's just stood in a corner. Zoe will correct me if I'm wrong here. It sort of stood in a corner. Any other cow got anywhere near it, it would charge out and chase them away. So, of course, that's sort of reflected in the numbers here, that it would approach a lot of cows, they wouldn't approach it, and when it did encounter them, they were always sort of desperately trying to get away from it because it was trying to attack them. So that sort of explains it. But the key thing here is by looking in a bit more detail about these interactions, not just looking at who's close to who, we can get a bit more interesting sort of dynamics of that social group. And again, the idea is here, this was just a very quick experiment. But if we look at the longer-term trial, where we've got 6 months worth of data, we haven't really started digging into that yet. But potentially, these sorts of changes in the social networks, or these very different sort of behaviors between approaching and leaving, that might indicate health or welfare. If a cow suddenly changes the way it interacts with other cows, that might be an indication that it's ill. So these are the sorts of things we're looking for in the data at the moment. So just to finish off, I asked the question, what about other animals as well? So this is actually from the Big Bang that Graham mentioned we took part in earlier this year. We took the system, we've got it running down here, we put the collars on, we didn't tell the kids to crawl around on the floor, they chose to do that themselves. But the point is, we could track humans just as well as cows. And in fact, we get a lot of interesting behavioural data on humans. And the guys who are working with OmniSense, I think Andy's in the crowd somewhere, one of the original reasons for using this system was actually in an old people's home to detect, for example, falls, or to detect whether the old person just wanders off so they can get them back. So it could work on tracking people. And in fact, if anyone's got a smartphone with them now, if we could get all the data from that smartphone, we'd have a very good record of your behavior. So think about that. Now, I'm not asking you for your data, but your phone company has that data. And if we can do all this sort of analysis and work out what you've been doing, they sure as hell can as well. So just think about that before you switch on all your data and let your phone company have it. But it could be, in all seriousness, it could be a useful thing, though, because, as I say, the potential health benefits, we could be monitoring people to see whether they're doing enough exercise, that sort of thing. Or we could look at how people behave in social situations. So this is something that we were doing. Again, Graham mentioned this. This is a zombie here, and there's another zombie there. We're looking at crowd behavior. So again, we can put these sensors on people and see how the crowd interacts as a whole. And Nikolai, who's in the middle there, we did a big project the last couple of years looking at how crowds might behave in evacuation situations. So this technology might be really quite beneficial there as well. So I think that is it. I think I'm only slightly late, so...